record that. Now I'm going to go back to the share. And go back to our slide. Sorry, we got okay. So was I able to answer your question? There's a video of the first two lectures, and I started recording this one, and those will be
Yes,
in module 1.
yes.
The McGraw-Hill assignments will cover some of the material we cover. They might also cover some material that we weren't able to get to. And I'll post a couple of quizzes with answers for chapters 4 and 5. Okay, so next we're going to talk about bacterial endospores. This is unique to gram-positive bacteria. They are the only ones that make endospores, not all gram-positive. Just some of them make endospores. This happens when they're in a hostile environment without nutrients and water, and enables them to survive. Now, life cycle in bacteria, when they're actively growing, we say they're vegetative. This is an older term, but it just means they're metabolically active. When the bacteria go into an endospore, they are no longer metabolically active. They're inert in a sort of a hybrid, I don't know, hibernation is not really the right term, but in a dormant state. And a lot of their essential things that they need are actually broken down, and they retain their genetic material. And then when they reach a favorable environment, they will sporulate. In other words, the spores will give rise to new actively growing bacterial cells. So these endospores, they make it very difficult to sterilize for them. Normal sanitation techniques will not destroy them. They're very resistant to heat, to drying, freezing, radiation, chemicals. Endospore formation starts when there's in the environment, there's a depletion of nutrients, especially carbon and nitrogen. When they reach a favorable environment, the sporangium will start sporulating, give rise to new cells. This takes about 6 to 8 hours, and you'll have actively growing cells again. So Clostridium and Bacillus are two of the genre. There are others within gram-positive bacteria that can make spores. So what will happen is the cell in a harsh environment would duplicate its genetic material, and when part of that replication will be in the spore, and the cell will slowly wall off the other genetic material, and you'll have this spore forming, and eventually this part of the cell will die off, and then in a in a more favorable environment, a new cell will start growing, and it will germinate through the endospore, and you'll have a new cell that can start growing and replicating. Germination begins for the spore under favorable conditions, exposure to water and a germination agent. Germination agent stimulates the formation of hydrolytic enzymes. So breaking something down with an enzyme, enzymes that carry out hydrolysis, what they do is they add a water molecule to break bonds. It's called hydrolysis or hydrolytic enzymes. They break down that cortex area of the endospore, and the core part rehydrates, starts taking up nutrients, and new bacterium grows. Once this process is initiated, it takes about 1.5 hours. And then, as we said before, within 7 or 8 hours, we have actively growing cells again. Diseases related to spore-forming bacteria, there's Bacillus anthracis, anthrax, Clostridium tetani, which makes the tetanus. This is a neurotoxin. Clostridium perfringens causes gas gangrene. Excuse me. And the Clostridium botulinum makes another neurotoxin called the botulinum toxin, which you've probably now in a lot of cosmetic ads, they advertise, they give very low amounts of this toxin to cause paralysis and straighten out wrinkles. If you remember, after the 9/11 attack, there was a lot of concern about anthrax being weaponized. And there was a vaccine for it that people in the military got. That was made by Emergent Biosystems here in Baltimore. It was one of the places that made the vaccine. The reason I mention that is now Emergent Biosystems, they actually have 2 locations in Baltimore. They're making 4 of the vaccines that are still in trials, actively making, and they have other locations in addition to Baltimore. One of the vaccines that they are making is the Johnson & Johnson vaccine. You've probably heard about that. It's just a single shot vaccine. It can be stored in a refrigerator up to a year. And they say at this point they have millions of doses ready to go. As soon as they get approval, they released the results and analysis of their phase 3 trial on Friday. Endospores are constant intruders where sterility and cleanliness are important. I said before, they are resistant to normal cleaning methods, boiling water, soaps, and disinfectants. Endospores frequently contaminate culture and media. Hospitals must protect against endospores and wounds. Destruction of endospores is important in the food canning industry. So, true or false, are plasmids vital to the metabolism of a bacterial cell? Anybody? Okay, back. Yes. True or false, plasmids are vital to the metabolism of bacterial cells?
True.
True.
They are not essential to the metabolism of bacterial cells. They carry extra genetic material that can make them more virulent, but in the plasmids in and of themselves are not necessary for the bacteria. They already have their single large DNA chromosome. Bacterial ribosomes are a little bit smaller and not as complicated as eukaryote ribosomes. The steps in endospore formation and germination. So first of all, when we talked about the gram stain, which group, gram negative, gram positive, make endospores?
The gram
Gram
positive.
positive.
Yeah, gram
Gram
positive.
negative.
I'm negative.
No, positive.
It's the gram-positive bacteria make endospores. Gram-negative do not. Some of the notable genre are the anthrax and the Clostridium that can make endospores. It starts with the cell being in a hostile environment. It duplicates its DNA and walls off one part of that DNA, and slowly the cell breaks down, and the one part that has the new genetic material, it will start forming an endospore, and rest of the cell will break down. And when it's in a nutrient-rich environment, it will germinate out and make a new bacterial cell.
What is vital to the metabolism of bacterial cells? Plasmids.
So
isn't-
in our next section, we'll go over bacterial metabolism. So some of the things that are vital is carrying out a process called glycolysis, and then aerobic respiration. Another important process that occurs in the cytoplasm is protein synthesis, which we'll talk about, the genetic code in another section, and how protein synthesis occurs. These occur in the cytoplasms of cells, and another important metabolic process is the ability to duplicate your genetic material. Does that answer your question? Okay.
We gonna
So
go over that, right?
Pardon?
We about to go over what you just was talking about, right?
We will, but not in this section. In the next section, we will talk about bacterial. We have a whole lecture on bacterial metabolism. And in a later section, we have a whole section about the genetic code, DNA replication, and protein synthesis. But all those are essential metabolic processes, and they are plasmids are just small pieces of genetic material, and they don't have the genes to carry out those processes. As I said before, they carry antibiotic resistant genes, carry genes for resistance to heavy metals. So 3 diseases caused by endospore bacteria. There's anthrax, caused by the Bacillus anthrax. They have a toxin disrupts, if I remember right, disrupts the cell membrane and causes lysis of the cell. There's 2 different species of Clostridium that both have neurotoxins, the one that causes tetanus and the one that causes botulinum. So we're going to talk a little bit about the differences between the archaea bacteria and prokaryotic bacteria. So the archaea are a unique group. We're not going to talk a whole lot about them, but they are important, and we'll discuss that. Most of the time, we'll be talking about the prokaryote bacteria and the gram-negative and the gram-positive bacteria. So the archaea are actually more closely related to us, to the eukaryotes, than they are to bacteria. They have ribosomal RNA sequences not found in bacteria. Their protein synthesis and ribosomal subunits are similar to eukaryotes. There are differences with other cell types. Their genetic information is only found in their ribosomal RNA. They have a unique method of compacting the DNA. I should say certain genetic sequences are found in the ribosomal RNA. They have unique membrane lipids, unique cell wall composition, and unique proteins for the pillin. If you remember, these are proteins that bacteria use for attachment. So comparisons of the three groups: the bacteria have a single, usually just a single, sometimes a few circular chromosomes. The archaea, it's always a single circular chromosome. In eukaryotes, our genetic material is linear DNA. Bacteria have a 70S ribosome. Archaea can have 70S, but the structure is more similar to 80S, like ours is an 80S ribosome. Their flagella in prokaryotes is hook rings and a hollow filament. Their flagella is a solid fimbrial-like structure. In single-cell eukaryotes, the flagella is a different protein and has what we call 9-2 microtubular arrangement. Contains unique ribosomal RNA sequences. That's true of all three groups. That's unique for the group. Protein synthesis is similar to eukaryotes. So actually in bacteria, it is, I'm saying this right, protein synthesis similar to eukaryotes, different in bacteria, but in archaea is more like eukaryotes. The presence of peptoglycan in the cell wall. So only the prokaryotes have peptoglycan in the cell wall. It is not in the archaea or in eukaryotes. The cell membrane lipids in bacteria are fatty acids with ester linkage. In archaea, they are long branch hydrocarbons with ether linkage, and eukaryotes with fatty acids with an ester linkage. Sterols in the cell membrane. Usually bacteria do not have sterols in their cell membrane. We mentioned the mycoplasma, a very small bacteria that has sterols in its cell membrane. Archaea do not have sterols in their cell membrane. Eukaryotes do have sterols in their cell membrane.
Can you remind me what the chemical composition looks like between the difference between the hydrocarbons and the fatty acids?
Well, hydrocarbons are just straight chain molecules, and we'd have to look at a picture. What you have in the, like, for example, the phospholipid bilayer of the cell membrane, you start with a glycerol, which is 3-carbon compound. The fatty acids are long chain hydrocarbons with a carboxyl group, which is COOH. And the glycerol has a OH group. And you remove the OH and form a bond with the fatty acid, you lose a water molecule. Typically, I think the long chain hydrocarbon fatty acids are somewhere around 18 to 20 carbons. Does that answer your question?
Yes.
So archaea are considered the most primitive of life forms. They probably more closely resemble cells that originated about 4 billion years ago. They live in habitats that are similar to extreme environments in the ancient world: heat, salt, in other words, high heat, salt, very acidic, pH, the air pressure was different. Many of them produce methanes. They are hypothermophiles. It means they thrive in very hot environments. Extreme halophiles means they thrive in very salty environments, and they are capable of reducing sulfur. So when we talk about metabolism, there are some bacteria that can use sulfur as an electron acceptor. Methanogens take CO2 and water to make methane. Common inhabitants of anaerobic swamp mud, bottom sediments of lakes and oceans, and even the digestive tracts of animals. The gas produced in swamps may become a source of fuel, but methane is also a greenhouse gas and can contribute to global warming. Extreme halophiles, they require salt to grow. Some of them can multiply in 36% sodium chloride that would destroy most cells. They exist in inland seas, salt lakes, salt mines, and in salted fish. They use a red pigment to synthesize ATP in the presence of light. Halophiles around the world. This is just an image of an area that's very salty. Psychrophiles, they grow at very low temperatures, and hyperthermophiles grow at very high temperatures. So they can thrive between 80 and 113 degrees. They cannot grow below 50 degrees. That would be the hyperthermophiles. They can live in volcanic waters, soils, and submarine vents found in the ocean. They are often very salt and acid tolerant. How are archaea cell walls different than bacterial cell walls? You remember that they share a similarity with eukaryotes. Well, actually, I'm thinking of the cell membrane, the sterols. The cell walls, if we go back here, bacteria have peptoglycan in their cell walls, but the archaea and eukaryotes do not have peptoglycan. We don't have a cell wall, but other eukaryotes like plants and fungi do have cell walls, but it does not contain peptoglycan. So the terms, we'll go over the definition. Methanogens are bacteria that are capable of taking CO2 and making it into methane gas. Halophile, phile means loving. They like halo, it refers to salt. They love salt. Psychrophile is cold. They love cold. And then there's the hypothermophiles. They love really hot temperatures. True or false? I don't think we mentioned this. Do they cause human disease? For the most part, archaea do not cause human disease. I don't on the test, I'll double check. I don't think I go in the classification, the bacteria and archaea. I didn't think this was as important to cover. So there's the two, we'll talk about these manuals, the Burgess Manual Systemic Bacteriology and the Manual Determinative Bacteriology. I think I'm going to skip this. I'll double check the test, but I don't think I go into this if you're interested in this, you can read over the slides. But they're important manuals for clinical microbiologists and researchers to identify unknown bacteria. The Burgess manual is comprehensive of bacterial and RKE relatedness, combines phenotypic information based on RNA sequencing. It's now available online. The determinative bacteriology is based entirely on phenotypic characteristics. So these are the, what you'd have to do is assay the bacteria in a clinical lab. And based on metabolism and so forth, you would determine what bacteria you have. It's useful for students of medical microbiology. There are 4 major divisions of the bacteria in RKE. I don't know if I'm going to say this right, because I've never used this term. Gracilocutes or gram-negative bacteria. The firmicutes are gram-positive bacteria. Those are the we were talking about. So generally, actually, I'm surprised there's another term used for gram-negative bacteria called bacteroides, and that's the term I'm more familiar with. There's the tourniquets, lack of cell wall. Those would be like the mycobacterium. And then the Mendus-cutes or the RKE. So this is the overall grouping. You're more likely to hear the term firmicutes for gram-positive and so some other important, oh, sorry, I'm due from there. Another important process in identifying bacteria, first, whether they're gram-negative, gram-positive, whether you have bacteria without cell walls, that would be like the mycoplasm, and then cell shape, arrangement, and importantly is oxygen usage. Some bacteria are aerobic like us, they need oxygen for metabolism. There's anaerobic bacteria that do not require oxygen for metabolism. Anaerobes means not oxygen. There are bacteria like E. coli that are facultative, so they can thrive in an anaerobic environment and switch over in an aerobic environment. These are just the groups of some medically important families and genre of bacteria. I'm just going to mention that, and there's several slides about them. It's not on the test. Species and subspecies in bacteria and archaea. Bacterial species, a collection of bacterial cells that share.
Will you
an
back up a
overall-
minute for me, please? So in lab, he's asking us to name the bacteria properly.
Are you going through this, the families and genre of bacteria? Is that what you're saying?
Yeah, no, so he asked that we identified whether something was a rod or a cocci or
So
but
would you prefer, because I didn't have this on the test and it's covered in your lab, but if you want me to go over these slides, I can.
not necessarily, just more about how you would name them, how you would say them appropriately.
Okay, well, let's start with bacteria with gram-positive cell structure. There's the Micrococcus KE, Staphylococcus, the Streptococcus, the family name, Streptococcus, the Peptococcus, the Bacillus. Remember, these are endospore, these are rod-shaped. So Bacillus causes anthrax, Clostridium causes tetanus and botulinum. Non-endospore rods, there's the lactobacillus, listeria, the propionum bacteria, they make propionum acid. The Crinibacteria is another spore-forming bacteria, can cause diphtheria. There's the Mycobacterium, which we mentioned before, that causes tuberculosis. They are not gram-negative or gram-positive. They have a thin cell wall and a thick lipo acids surrounding them that makes them very resistant to antibiotics. There's the Neisseria. These are gram-negative bacteria, cause meningitis. The Tinobacter, which is an aerobic bacillum. There's an anaerobic cocci. Actually, I'm not familiar with this one. Veillonella, probably not saying that right. If you do have trouble with the pronunciation, sorry, my dog seems to want to come in. Go to YouTube and or you can Google pronounce, type in the word, and it'll usually give you a YouTube video of somebody pronouncing it. Pseudomonas is an aerobic rod. They are a real problem in pneumonia and burn infections. Legionella's disease is another one. Okay. Facultative or anaerobic rods in Vibrios. There's E. coli, Edwardsia, Citrobacter, Salmonella. There's a species of Salmonella that causes typhoid fever. There's Shigella, which is a bacteria that causes severe dysentery. Klebsiella, Enterobacter, Serratia, Proteus, Yersinia. Yersinia pestis is the cause of the bubonic plague. Vibrio.
I think my question was a more simple than what you're answering. Like when you say E. coli.
So we mentioned before, sometimes when you're referring to a bacteria, a lot people will, instead of saying the genre, they will just say the first letter. So like Vibrio cholera, you might just say V. cholera, or Salmonella typhi, you might just say S. typhi.
Okay, that's what I was asking.
Okay, sorry. Do you want me to continue?
Yeah, well, no, not necessarily if it's not on the test, but I did note on there that there was a difference between the gram positive and the gram negatives that we're trying to identify in labs on
That's
those slides.
what's in the bacteria, what's really important is knowing the difference between the gram negative, gram positive, and the microbacterium that don't fit into either group. And then remember, the mycoplasms do not have a cell wall, and they're unique that they have sterols in the cell membrane like eukaryotes do. The other important things are the shapes, whether it's a coccus or a bacilli, which is rod-shaped. Coccus is the spherical shape. And then you have the odd ones like the Vibrio, it's kind of a bent shape. The other important things are whether they're aerobic, anaerobic, or facultative anaerobes. So bacterial species would be bacterial cells that share an overall pattern, similar traits. Subspecies are strains or types. So those are bacteria of the same species, but they might have a really unique characteristic. A lot of times in bacteriology, we're talking about a special virulence factor that a subspecies might have. A serotype is a species that stimulate a distinct pattern of antibody response due to distinct molecules they might have on their surface. So there were the two manuals. One is more clinical, the determinative manual, and the other one will talk about evolutionary relatedness. That's something you'd have to do in sequencing. A lot of times to look at evolutionary differences, we sequence the RNA patterns in ribosomes because they are the most stable. Okay, let me see where we are time-wise. So it's 8 o'clock. What I want to do is call roll for people who came in late. Is Brittany here?
I'm here.
Okay. Is Kelly Butler here? Kelly Butler? No. I'm going to spell this. I don't want to mispronounce it. Ayelech. Last name D'Amica. You're not here? Yes. Dockett, last name Dockett. Are you here? Ellis, is Ellis here?
I'm not Joy.
Pardon? Was that person talking Tanisha Ellis?
No, I don't know who that was, but I'm here.
Okay, good. Catherine Fuentes,
I'm here.
thank you. Janae Hamlin. Hamlin,
I'm here.
okay, thank you. Is Jamal Howard here? No. Jeremiah Kelly. Jeremiah Kelly. Brian Kincaid.
I'm here.
Okay, good. Jeremy Litton. Okay. Kiara Lucas. Actually, I thought I saw you. Kiara Lucas.
I'm here.
Okay, good. Lillian. Is Lillian here? Last name Nwagbara. Not here. Elizabeth Salami. Is Elizabeth here? Okay, so let's just take a quick break for a few minutes. And
Lillian is here, Gbara.
I'm sorry, who was that?
Ngbara. Lillian is here.
Okay, thank you. So I'm going to stop sharing this, and when we come back, we will start with our final chapter. System 1.
Professor, this chapter is like 84 slides. Are we going to be able to make it through?
If we still have 90 minutes. If we don't make it through, whoops, I'm sorry, if we don't make it through, I won't include the last slides at the end.
Can you hear me? Thank you.
But I'm just going to take a break for just a few minutes, and we will start on chapter 5. Come on. You might have to call the dog and see if he can go outside or something. I got about another 90 minutes. Okay, so before I start with chapter 5, I just want to go over it next week is exam 1. Exam 1 covers chapters 1, 3, 4, and 5, does not cover chapter 2. In the McCraw-Hill online learning assignments, there is a smart book and there is a quiz that you can take. You can read over the PowerPoints and take those if you want some extra credit. I said at the beginning of the class, in the McCraw-Hill, there's about 30, I believe 31 assignments. You need to do 24 of those assignments. In other words, 12 smart book and 12 quizzes. That'll get you to 150 points or 15% of your grade. But you can do as many of those assignments as you want. I've had students get 160, 170 points by doing extra assignments. They are all due May the 15th. In other words, the Saturday after our final exam. What else? I will set up a Zoom meeting when you take the test next week, but I will just monitor it if people have questions. I will decide later. I might just lecture for 1 hour to get started on our next session. You'll have about 70 or 75 minutes to do exam 1. So once you start, you got to complete it, but I'll open up open the test up for the entire class period. In other words, the class period's 2.5 hours, but you have to, once you start the test, you have to finish it before 9:30, because that's when the test will close. Okay, so this chapter now is going to focus on eukaryotic cells and microorganisms. I believe we'll also talk about helminths, which are the parasitic worms.
Hello, Professor. I have a question.
Yes.
I didn't understand what you mean that you're gonna about the Zoom. Is it the Zoom for the exams or what?
So you will not take the test through Zoom. I don't even have to have the Zoom going on for the test. The reason I'm going to open up a Zoom meeting is if case somebody's having an issue, they can jump over to Zoom and they can like send me a message in chat if they have an issue or a question.
So you mean the exam is gonna be is going to take the same day we are taking the same day of class or what?
You will take, so you're required to take exams during regular class time. So I will open up the test for between 7 and 9:30 next Tuesday. And you'll take it through the McCraw, you'll see it and you'll go through just like you would if you're doing an online assignment. You'll see exam 1. You can't see it right now. I haven't posted it, but it'll be made available and it'll only be open from 7 to 9:30 and you'll have around 75 minutes to take the test. So you don't want to start later, like if you start at 9 o'clock, you'll only have 30 minutes to take the test. So you want to start it early. And, but you have a little wiggle room. You could obviously start at 7:30 if you want. And I'm contemplating, I might put a lecture up for an hour, but you do not have to attend the lecture. And I will record it and put it in the module.
All right, thank you, sir.
Okay.
I had a quick question. I was wondering if you were going to still put the slides up that was focused on the test.
Yes, I think I mentioned that before. I have some just notes that mention the slides for the exam. I'll get those put up by tomorrow. Okay, so we're going to talk about the history of eukaryotes. We've already talked a bit about similarities and differences, bacteria, the archaeobacteria and eukaryotes. Talk about evolution, what they refer to as the last common ancestor. We're going to talk a little bit more detail about the structure of eukaryotes, talk about between unicellular and multicellular. So eukaryotes can be single cell, also called unicellular. Eukaryotes mostly are multicellular. So if you remember, the protists are single cell. Bacterial cells and archaeobacteria are always single cell. In other words, they are always unicellular, same synonymous terms. So we believe the first eukaryotic cell appeared on Earth 2 to 3 billion years ago. Bacteria and eukaryotes evolved from a predecessor called the last common ancestor. Whatever this ancestral cell was, it was neither pro or eukaryote, but it gave rise to bacteria, archaea, and eukaryotic separately. Organelles originated from more primitive cells that became trapped in other cells to become eukaryotic cells. So about 3 to 4 billion years ago, they believe there was a common ancestral cell. Its genetic material was probably RNA. And that cell gave rise to archaea bacteria and what we call the prokaryotes. We believe what happened is that a symbiotic relationship occurred. Endosymbiosis is when a symbiotic organism lives inside another organism. And the reason we think that is in modern day eukaryotes, the mitochondria are that we have in our cells is very similar to bacteria. And the chloroplast in plant cells is very similar to photosynthetic bacteria. The first primitive eukaryotes were single cell and independent living. They become specialized to perform particular function. In other words, they lived in colonies. Complex multicellular organisms evolved when cells lost the ability to survive apart from a colony. Eventually, these became more specialized to form tissues, and the tissues then formed organs, and we got more complex organisms. So we've already mentioned the protista or protozoa. They are always single cell. These are eukaryotes. The fungi and the algae can be either single cell or multicellular. So yeast is a fungi, single cell. Mushrooms are multicellular, for example. Multicellular organisms, we mentioned we study helminths, which are small parasitic worms. in their reproductive state, you would actually have to observe them with a microscope in the egg or larval forms. When they form a parasitic worm, those are visible to the eye. We believe that eukaryotes and prokaryotes were evolved from a common ancestral cell. Tissues and organs. Well, organs are made of tissues. So for example, you have hepatic cells that can form tissues, can be part of the liver, but obviously you have other tissues within the liver. You have your circulatory system, which are different type of tissue and different types of cells. 3 types of eukaryotics studied by microbiologists. Well, we just mentioned that. The protozoan, the fungi and algae, and the parasitic worms. We'll talk about the flagella structure of bacteria, archaea, and eukaryotes that's different. Talk about the glycocalyx characteristics of eukaryotes. Remember, in bacteria, they have a, I'm thinking of spore. They have capsules, or one type of capsule. It's made of sugars in bacteria, but there's a different one in eukaryotes. There are some eukaryotes that have cell walls. I mentioned before the fungi and plants have cell walls, but it's made of different material. It's not made of peptoglycan. We've already mentioned some differences and similarities between between eukaryotes and bacteria, and differences we'll talk about the differences in their membranes. So features of eukaryotic cells: they all have a cytoplasmic membrane. They have a membrane-bound nucleus. They have a mitochondria. They have an endoplasmic reticulum where protein synthesis occurs. A Golgi apparatus. They can have vacuoles. A cytoskeleton made of actin proteins, and they can have single cell ones can have a glycocalyx. Some eukaryotes can have cell walls. They can have appendages for movement like flagella. They can have chloroplasts. Obviously, the plants and algae have chloroplasts for photosynthesis. A generalized eukaryotic cell, if it's a protist, it could have flagella, but it's made of different proteins than found in bacteria. They have a mitochondria, which is necessary for aerobic respiration. Golgi apparatus for these vacuole can form. A notable one is a lysosome that we'll see in phagocytic cells talk about. Mitochondria, again here for aerobic respiration. The nucleus has the genetic material with a nucleoid region, pores to allow the message RNA to come out. In the endoplasmic reticulum, the proteins can be modified and sugars can be added to them. The centrioles are necessary in DNA replication. Plants and fungi have a cell wall. So in plants, the cell wall is made up of cellulose. In plants, it's made of chitin, the same material found in the exoskeleton of invertebrates. Rough and smooth endoplasmic reticulum. When the ribosomes attach to the endoplasmic reticulum, it's called rough endoplasmic reticulum. What else? They can have these actin filaments that help give the cell structure. So eukaryotic flagella is thicker, more complex, can even be covered by an extension of the cell membrane. They have these long sheaths, cylinders, hollow microtubules that are 9+2 arrangement. So if you took a cross section of flagella in a eukaryote, you would see 2 tubes here, and then you see this arrangement of 9. So that's why they call it the 9-2 arrangement. Cilia, so the single cell eukaryotes, the protists, they can have, for movement, they can have this cilia that we actually have in our respiratory epithelia. And it can beat and enable the cell to move, or in some instances beat so they can get food in or expel waste products. Glycocalyx is an outermost boundary, also called an extracellular matrix. So we actually, in us, we've retained this. In our cells, we have outside of our cells something called an extracellular matrix. It's made up of various polysaccharides, has a network of fibers. In protists, they can have a slime layer and a capsule. Cell walls, I mentioned, is found in fungi and algae, and also in plants. In fungi, it's made of chitin. The cell wall is rigid and provides support and shape, different chemicals. It's a different chemical composition than bacterial cell walls, so they do not have peptoglycan. Chitin or cellulose in fungi. So this would be the cell membrane, the chitin, some glycoproteins, mixed glycans, and then they can have this other layer on. This is going from inside the cell to outside. They can have this glycocalyx. The cytoplasmic membrane can be a typical bilayer of phospholipids embedded with proteins. That would be true of fungi, true of us. It contains sterols, which are ring structures like cholesterol. Sterols help stabilize eukaryotic membranes. Eukaryotic membranes are selectively permeable. They determine what gets into or in and out of a cell. They can have sophisticated mechanisms for transporting nutrients in and waste products out. So the eukaryotic flagella is made of a different protein. They can have some cell membrane around them, and they have that 9-2 structure that flagella and bacteria don't have. Cilia can be for locomotion in single cells. In the beating, the cilia can allow them to take in food. Glycocalyx is for protection. They mentioned what the other functions were. I don't believe with the outermost boundary, that glycocalyx is what comes into direct contact with the environment. So eukaryotic cell walls are a different composition than prokaryotic cell walls. Prokaryotic cell walls, whether it's gram-negative or gram-positive, we mentioned before, it's made of peptoglycan, which are 2 modified sugars that are in repeating units. These are not found in eukaryotes. In eukaryotes, the fungi have chitin, plants have cellulose as their cell wall. So we're going to talk about, just check the time, okay. The main structural components of a nucleus. We're going to talk about the nucleus region, the endoplasmic reticulum, Golgi apparatus, how they're involved in transport. We're going to talk about the mitochondria, the importance of ribosomes and the differences between bacterial and eukaryotic ribosomes. We've already mentioned that before. The bacterial ribosomes are the 70S ribosomes, and ours are 80S for sedimentation coefficient. And our ribosomes are more complex. They have more proteins and RNA in them. And we talk about the fibers of the cytoskeleton. That's what gives cells their structure. And we're going to talk about endosymbiosis. So symbiotic relationships between two species. Endosymbiosis is one species lives inside another species and actively replicates on it. So the bacteria in our gut are not endosymbionts. They don't live inside our cells. They just live inside us, if that makes sense. A true endosymbiont would live inside a cell. They even found recently a sponge, which is actually an animal, had developed a symbiotic relationships, I believe, with algae that lived inside their cells and carried out photosynthesis and provided nutrients. Rather unique endosymbiotic relationship. So the nucleus region in eukaryotes, it's compact. It's separated from the cytoplasm. It's surrounded by a membrane called the nuclear envelope. Within that chromosome material, there's a nucleosis region, stains intently due to the active synthesis of RNA. So it's a site of ribosomal RNA synthesis in the chromosome. So if you looked at this nucleus and the nuclear membrane, in the region where the DNA is, in this dark staining region, the nucleus, this is where the cell's actively transcribing RNA. Chromatin is the material of eukaryotic chromosomes. It's the unit of genetic information. In bacteria, they have a single circular chromosome. In us, we have long linear DNA. We also have proteins that bind to our DNA called histones. Bacteria do not have histone proteins. The chromosomes become visible during mitosis. So this is each time a cell replicates. So it goes through these different stages. It duplicates the genetic material. You have these centromeres and formation of spindle fiber that enables the cell to separate. The chromosomes line up along a mid-region of the spindle fibers and then separates. Remember, the chromosome is duplicated, so now you have two daughter cells and they separate and the whole process starts over. So this is mitosis. So the production of sex cells is meiosis, and you don't get a complete, you don't wind up with a double chromosome. You wind up with a reduced, you wind up with just one strand, and then you form a complete cell, a gamete, to replicate. The endoplasmic reticulum are microscopic tunnels involved in transport and storage. The rough endoplasmic reticulum is where it's covered with ribosomes and proteins are made, and then the proteins in the endoplasmic reticulum are packaged and transported. They can be modified, sugars can be added to them. Smooth endoplasmic reticulum just does not contain ribosomes. So the endoplasmic reticulum is just a closer look. It will be directly connected and interact with the nucleus. So proteins can be synthesized. So the genetic material gets transcribed into RNA. The RNA goes on to the ribosomes to make proteins. You can see this is a very dense active site. This would be the nucleus region with the DNA. This shows you the rough endoplasmic reticulum covered with ribosomes. And this is where active protein synthesis is occurring. The ribosome binds the message RNA, and you have synthesis of the proteins, which is we will talk about that in more detail in our genetics section. So the Golgi apparatus is a site of protein modification and transport. The Golgi apparatus is a stack of flattened disc-shaped sacs. It's closely connected to the endoplasmic reticulum, so the proteins, once they're synthesized, can go to the Golgi apparatus and be modified for transport. Where the two are in close contact, the ER forms transitional vesicles that then go over into the Golgi apparatus. The Golgi apparatus can add polysaccharides and lipids to the proteins, and the transitional vesicle pinches off the vesicles. I think I improperly said that the modification of newly synthesized proteins occurs in the endoplasmic reticulum. So sorry about that. It's actually occurring in the Golgi apparatus. So some of these vesicles are lysosomes, and some are transported as secretory vesicles that are then leave the cell. So this shows you, like over here, not shown would be like the nucleus. You have synthesis of the proteins. Proteins could be transported in vesicles to the Golgi apparatus where they can be modified. So genetic information originates from the nucleus. Proteins are synthesized on ribosomes, deposited into the endoplasmic reticulum. Proteins are then transported to the Golgi apparatus to be chemically modified and packaged into vesicles. So this shows you the whole process, the nucleus region, transcription of ribosomal RNA, protein synthesis occurring, the proteins being transported to the Golgi apparatus where they can be modified, and then they can be in these membrane-bound vesicles that can go to the cell membrane. And you can have a secretory vesicle where something is just secreted, and after it fuses, it can open up and release whatever these products are into the outside environment of the cell. So some of these cellular vesicles are lysosomes that originate from the Golgi, contain a variety of enzymes. These will often have enzymes that are involved in intracellular digestion of food or protection against invading microorganisms, which we'll see again when we have our immunology section. Vacuoles or membrane-bound sac, they contain fluids or solid particles to be digested, excreted, or stored. The mitochondria that's found in our cytoplasm is a double membrane structure. We believe it originated from a common, an ancient prokaryotic cell that then lost its ability to live independently. So the mitochondria supplies the bulk of energy for our cells. We'll talk about that in our next section about bacterial metabolism. It has two notable structures, a cristae, which folds in the inner membrane. This holds the enzymes and electron carriers for aerobic respiration. And then the matrix area. It has the chromosome, the mitochondria has its own chromosome, its own ribosomes, and it replicates independently of the cell. So, so we said that before. So mitochondria divide independently of the cell. They have a single circular chromosome like bacteria do. Their ribosomes are similar in size to a bacterial ribosome, which are some of the reasons we believe that our mitochondria resulted from an endosymbiotic relationship. Photosynthetic plants have chloroplasts, which we also believe resulted from an endosymbiotic relationship. They have their own ribosomes like bacterial 70S. They have a double membrane similar to what bacteria have. And it is the chloroplasts where photosynthesis occurs, primary producer of organic nutrients, and is where we get oxygen in our atmosphere. It comes from the metabolism that occurs in the chloroplasts. So the ribosomes are distributed throughout the cell. They're scattered freely in the cytoplasm and the cytoskeleton of the cell. I mentioned before, they're attached to the rough endoplasmic reticulum. Mitochondria and chloroplasts have their own ribosomes, their own genetic material. Chains of ribosomes hooked together, or actually it's multiple ribosomes hooked to a message RNA are called polyribosomes. Eukaryotic ribosomes are similar to bacteria ribosomes in their structure. There's both a large and a small subunit. In eukaryotes, that's referred to an 80S, made up of a 60 and 40S subunit. In eukaryotes, the cytoskeleton has important functions. It helps to anchor all these different organelles found in the cell. They allow for moving of the RNA and vesicles inside a cell. They permit shape changes and movement for the cell. The cytoskeleton is made of actin filaments, intermediate filaments, and also microtubules. Major elements of life in each in each organism type. So if we compare bacteria and archaea cells versus eukaryotes and viruses, all have genetic material. A true nucleus is lacking in the archaea and the bacteria. Viruses have no nucleus. Only eukaryotic cells have a nuclear envelope. Viruses are not considered cellular. They do not undergo mitosis. We undergo mitosis, cell division. In bacteria, it's called binary fission. Bacteria don't exactly produce sex cells, though we'll talk about how they exchange genetic information. Viruses do not at all produce sex cells. They do not undergo mitosis. Biosynthesis, occurs in both bacteria and eukaryotes, but bacteria do not have membrane organelles like Golgi and endoplasmic reticulum. Viruses do not carry out biosynthesis. They do not have ribosomes. Photosynthesis, some eukaryotes carry out photosynthesis, some bacteria can carry out photosynthesis, viruses can't. Flagella, some bacteria have flagella, single cell eukaryotes can have flagella, viruses not. For, sorry, did I skip? Yeah, flagella. Cilia is found in eukaryotic cells, not in bacteria, not in viruses. The cell membrane, viruses can have a cell membrane, but they take it from the host cell that they infect. Viruses do not have a cell wall. They do not have a capsule. They do have a protein capsid that their genetic material's in. Size differences, obviously eukaryotic cells are much bigger. Viruses are even smaller than bacteria. So eukaryotic chromosomes are visible during all portions of the cell cycle. Is that true or false? That is false. It's only during replication that you can see the chromosomes. The blank endoplasmic reticulum is responsible for packaging and transport of proteins. That would be the rough endoplasmic reticulum. Vesicles like the lysosomes, digestive enzymes. I'm sorry, somebody had a question?
No, I was just answering the third one, lysosomes.
The third one, yes.
Yeah.
True or false? I'm sorry, I'm jumping a bit ahead. True or false? Mitochondria and chloroplast contain 70S ribosomes and circular DNA. Is that
False.
true or false?
False.
That is actually true. Remember, we believe that mitochondria and chloroplasts evolved from an endosymbiotic relationship, that originally the mitochondria and chloroplasts were prokaryotic cells living independently and developed an endosymbiotic relationship, and they've lost their ability to live independently.
Excuse me, Professor, I'm sorry. I know we've mentioned this quite a few times, but the S, what does this mean? Like the 70?
It was, I believe it's not a unit of weight. It's a unit of sedimentation. So when they have to use these very ultra centrifuges, high-speed centrifuges to isolate ribosomes when you break open a cell and you're trying to separate out the different parts.
Oh, like with PCR?
No, PCR is not a separation procedure. Polymerase chain reaction is a procedure to take RNA and convert it to DNA and amplify a sequence.
Mm-hmm. But it's separating it out by weight, though, right?
Well, yeah, but the units they use are as I remember, is the sedimentation coefficient. 70S is not a unit of weight, because if you remember how the small and big ribosome units come together, it's not additive. It's
Okay.
how they separate when they're centrifuged.
Okay. Thank you.
So we're going to talk about fungal anatomy in this section: heterotrope, saprophobe, and parasite. We're going to talk about fungal hyphae and the mycelium. We're going to describe two ways fungal spores arrive We're going to list 2 detrimental and 2 beneficial activities of fungi, obviously from our viewpoint. Where are we? Slide 36. So there are approximately 30 to 4 million species of fungi. There's the macroscopic fungi. We can see those, the mushrooms, puffballs, gill fungi. I believe that's the same thing as bracket fungi. You see them growing on trees. The microscopic fungi are the molds in yeast. So yeast cell, it is round to oval. It uses acellular reproduction. It is single cell fungi. You can get these long thread-like cells found in filamentous fungi or molds. And you can get pseudohyphae, which is just the chain of yeast cells. Some fungi can take either form. We call that dimorphic. Morphology refers to shape. Di would be 2 different types of shapes. So penicillin is a mold. And also non-separated hyphae. I was showing you the 2 differences here. These are able to form a septa and separate out, as in penicillin. And this is another mold called Rhizopus that doesn't have these septa where they can separate from each other. Here's an actual micrograph showing you the formation of the septum of the hyphae, which are the long branching molecules of, I'm sorry, cells of fungi, not molecules. So the microscopic morphology of yeast, they are a eukaryote. So they have a nucleus, they have mitochondria, they have Golgi apparatus. They have a cell wall, which is made of chitin. They have ribosomes like our ribosomes. One way they can reproduce is they can just bud off a new cell, replicate the genetic material, and this will just bud off, and they will just keep doing that. You can see a new fungi cells here budding off of these cells. Fungi, they can be heterotrophic. That means they can require nutrients from a variety of substrates. Saprophobes, they are the fungi that you find on things that are dead and dying. So they basically, dead plants and animals, they will live off of that. Some fungi are parasites. They either live on bodies of living animals or plants. So if you ever had athlete's foot, that is parasitic fungi living on your skin. Oh, and they had a picture of it. Forgot about this. So it's obviously a warm, moist environment. And the fungi are actually growing on the surface of the skin. Here's a close-up picture of that. Most microscopic fungi grow in loose association or in colonies. So the mycelium is a woven, intertwining mass of hyphae that make up the body or the colony of a mold. The septa, remember, we refer to as cross walls that the hyphae can divide and break into segments, or they can form spores, which are reproductive bodies. This shows you a bread with a typical mold growing on it. So they can form these hyphae that can branch about. They can form These reproductive hyphae form these sacs that will release spores, and the spores will go in the environment, and they can start the whole replication over. The spore can form the hyphae can branch and grow, and then they can form, I think it's called a sporangium, and they release spores into the environment. Fungi have many complex and successful reproductive strategies. Simple outward growth of the hyphae, those branching areas that you saw. Fragmentation, separated pieces of mycelium can generate a whole new colony. Or the primary reproductive mode is spore formation. Fungal spores should not be confused with the resistant non-reproductive bacterial endospores. So fungal spores are different from endospores. The sporangiospores that we saw there formed at the end of the hyphae. They form a sporangiospore, a sporangium, and they can release free spores to continue the replication. Or these hyphae can break off. They can form the branching hyphae, and they can continue the replication of the fungi.
What's the hyphae again? I'm sorry.
So in the life cycle, there's the fungi can replicate two different ways. So let's start here. You have a spore, and it gives rise to some more fungi, and it forms this branching hyphae. And these can keep growing. And one way they can replicate is they'll eventually form a reproductive sporangium here at the end that will then release spores. So that's one way of replicating.
Is it a fungus or a?
This is a fungus. This is
No, the hyphae is a fungus.
all of these structures are the fungus.
Okay. Thanks.
And the spores are the reproductive part of the fungus. So what you're seeing here, like on the bread mode, this is all the branching hyphae. And if you had a microscope, you might see some of these structures, and these are actively releasing spores. So they can just continue to grow and branch as the hyphae, or they can eventually release spores into the environment. So, okay, so the spore formation is asexual, excuse me, asexual. And the sporangiospores is what this structure is that releases the spores. Or you can have free spores that are not enclosed in a spore-bearing sac. So you have two different ways this can occur. Sexual spore formation is different than that process. So basically fungi can form this way, which is an asexual spore formation, or they can replicate this way. So important variations occur when fungi of different genetic makeup combine their genetic material. Slight variations in form and function are potentially advantageous to the adaptation and survival of the species. Sorry, my mouth gets really dry after talking for 2 hours. Identification in medical specimens. So the fungi can be isolated on special media, observed microscopically and macroscopically. Asexual spore-forming structure and spores can be used to identify genus and species of fungi. The hyphal type, colony texture, the color, the physiological characteristics. All of these can be used to identify the fungi. So these are actual pictures of the fungi. The one with the spores in the sac and the one with the spores are actually not enclosed in a sac, being released into the environment. And these are the branching hyphae of the mold. So there are some medical conditions caused by fungi. Most human fungal infections occur through accidental contact in soil, water, or dust. Pathogenic fungi are community-acquired infections caused by environmental pathogens. Hospital-associated infections are by fungal pathogens in clinical settings. Opportunistic infections caused by pathogens infecting already weakened individuals. So an opportunist infection is somebody who's immune compromised or they are already sick and they can get another infection. Other medical conditions caused by fungi, people can have allergies to fungal spores. Neurological conditions can happen because the fungi can make a toxin. On the other hand, there's beneficial impact of fungi. Obviously, we use yeast to make bread and alcohol. Fungi are important in decomposing organic matter and returning nutrients and minerals to the soil. Very importantly, in plants, fungi have a symbiotic relationship with plant roots that help them to take in water and nutrients. Fungi can make antibiotics. We already mentioned alcohol. They can make organic acids and vitamins. They can be used in food flavoring. The alcohol, beer and wine, the gas that causes bread to rise is all due to the presence of fungi. So a blank type of fungi is a type that derives nutrients from dead or decaying material. This is a saprophore. True or false? Fungal spores have a tough outer coating that allows them to survive in extreme conditions. And that would be the description of an endospore from bacteria. So blank or asexual spores contain a sac-like structure that is a sporangium. Pathogenic fungi require a host to complete their life cycle. That's not necessarily true. They are taking advantage of a situation to reproduce. Let me see where we're at. I think we have about 30 slides left. So protists are the single cell eukaryotes. Note the protozoan characteristics illustrate why they are informally placed in a single group. There are three ways of locomotion by protozoan. We'll talk about a cyst stage, which is a part of the reproduction of the protozoan. We'll talk about some human disease caused by 4 types of protozoa. Algae and protozoan have been traditionally combined in the kingdom Protista, which is made up of subkingdom algae and a subkingdom of protozoan. The protists are unicellular eukaryotes. They can form colonies. They lack true tissue. Some protists, like the algae, are photosynthetic. Seaweed and kelp are the most easily recognizable forms. Algae exhibit all the eukaryotic organelles. They have chloroplasts that contain green chlorophyll. They can also have some other chlorophylls, yellow, red, and brown, give them yellow, red, and brown coloration. Algae are widespread inhabitants of fresh and of marine water. Plankton is an algae. It's a floating community of microscopic organisms. It's essential in the role in the aquatic food web and produces most of the Earth's oxygen. Primary medical threat of algae is ingestion of toxins during a red tide, which is a type of algae. The protozoa, there's over 65,000 species. Most are harmless inhabitants of water and soil. A few species are parasites and responsible for hundreds of million infections in humans per year. Protozoans are single cell containing major eukaryotic organelles, but they do not have chloroplasts, which are found in algae and plants. They can have organelles that are highly specialized. They can have mouths and digestive systems and reproductive tracts and something resembling legs for locomotion. Heterotroph means a variety of different foods. They require a complex organic forms. They are free living species. They can scavenge dead plants and animal debris. They can graze on live cells of bacteria and algae. There are parasitic protists that live on fluids of the host, such as plasma and digestive enzymes, and they can actively feed on tissues. They can move by these false feet called pseudopods, pods for feet, pseudo obviously false. These are amoeba. They can also serve as feeding structures. They can have flagella. That's a different structure than what's found in bacteria. They can also have cilia over their entire surface. 4 types of locomotion, with the membrane actually an undulating membrane with a flagella. You can have an amoeba, for example, with these pseudopods. It constantly changes its shape. You can have these protists with cilia on the edge here. Actually, it doesn't describe this one very well.
It's a sporosome.
I don't know, I don't see a description of this one.
It's a sporosome that has no specialized locomotion organelles.
So it would actually be non-modal. In other words, it can't move. This has pseudopods, the cilia, and this one has the flagella, and this would be a non-mobile or motility, non-motile organism. So protists can have a feeding and dormant stage. So the trophocyte is the motile feeding stage. It requires food and moisture. Under a harsh condition, they can form a cyst, which is dormant, a resting stage. It forms when conditions become unfavorable. It's an important way they can spread disease. It is a resistant structure, though not like an endospore, but it is a dormant stage that can help spread the disease. So the active, the trophozoite, the active feeding stage, and then if it's in a harsh environment, it can form a cyst, and the cyst can survive inside a cell, and then eventually start to replicate and break out of the cell and become an active form again. Life cycles of protists can vary from simple to very complex. Some protozoans exist only in the trophocyte stage. Many types alternate between the active feeding stage and the cyst stage. Their life cycle dictates how they are transmitted. Protozoan reproduction can be simple, asexual, usually they just replicate by mitosis. Several parasitic species reproduce by multiple fission. Some sexual reproduction can occur. They can carry out conjugation, which is the exchange of genetic material, which we will see in our section about bacteria. They can also carry out conjugation, some species of bacteria. They don't exchange all their genetic material, just some of it. So protozoans, they can have a single flagella or various flagella. They have a single nucleus, sexual reproduction by syngamy. They can form cyst or they are free living. Those using amoeba motion, which is the pseudopods, they have asexual reproduction by fission, usually unicellular. I'm sorry, my mouth is not working. Mostly uninucleate, so they have a single nucleus. They can carry out fission, which is asexual, which also occurs in bacteria. But this is unique in some, not in the amoeba, but some protists, they can have two nucleuses, but not in the amoeba-like. These will usually have a cyst stage, and they can be free living, but not infectious. Classification of selective protozoan using cilia to move. Some have cilia and tufts. Most can develop cyst. They have a macronuclei and a micronuclei. Division by transverse fission. Most have a definitive mouth and feeding organelle. They are free living and harmless. Those with no motility. So remember, we had our 4 groups: cilia, fungal, and the pseudopods, and then the group that has no motility. Motility absent in most of these sporozoa. They have complex life cycles with well-developed asexual and sexual stages. This entire group is parasitic. Most protozoan can be identified to the level of genus because of their unique appearance. Considerations and identifications: shape and size of the cell, type and number and distribution of locomotive structures, presence of organelles or cysts, number of nuclei, blood, septum, cerebral spinal fluid, fecal, vaginal specimens smeared on a slide can be used to identify them. So in parasitology, some important protist parasite, Entamoeba histolytica, is an intestinal parasite. Its reservoir is humans, water, and food. Nagligeria and I'm probably not going to say this right, Anthramoeba. They can cause brain infections. They are found free living in water. Ciliated protozoan, Blantydidium coli. This is not a bacteria, remember, it's a protozoan. It also causes intestinal infections found in pigs and cattle.
I have a question.
Yes.
Are all parasites harmful?
Yeah, the definition of a parasite is something that causes some kind of harm to the host. That is just the basic definition. So bacteria could be considered a parasite, some fungi parasites, protozoan, there's some protozoan that are parasites. To be honest, I'm going to look over this. I don't think on the test, I had questions about the protozoan pathogens. So at this point, with not a lot of time left, I'm going to, if you're interested in the pathogens, Giardia, it's a little more common than you think. My dog got it from drinking stagnant water, and they have to take a special medication to get rid of it. He started becoming lethargic and started eating less. And the vet just said he was somewhere drinking. When we went for walks in the woods, went on hikes, he was drinking stagnant water and picked up this parasite. So I'm going to skip over this. I will double check, but I don't believe this is on the exam, though there are some important ones in Africa. Trypanosome causes sleeping sickness in Africa. There's another type of trypanosome that causes Chagas disease, which is endemic to South and Central America. Long crescent-shaped cells with a single flagella. They are found in the blood during infection. And also there's insects that can carry them. Blood-sucking insects can transmit them as the vector. So algae don't cause any type of human disease. Is that true or false? That is actually false. They can. There was actually in the Choptank River in Maryland a while back a Pfiesteria outbreak of algae, released a toxin, and people that were fishing in the water got sick from it. The 3 ways that protozoan can move is cilia, formation of pseudopods, or flagellum. True or false? All protists have a trophocyte form, but not all produce cysts. Is that true or false? That is true. They all have to have a living, feeding life cycle, but not all of them produce cysts. The blank term is most often used to describe protozoan and helminth pathogens. Somebody just used that term a second ago.
Parasite?
Yeah, parasite. So what do we have here? I think we have 10 slides left, maybe. I don't believe the helminths. I'll double check the test. I'm not sure. I don't think let me put it this way. If there's questions about protozoan pathogens or helminths, I'll mention what slides those are on. The helminths are the parasitic worms. There are the tapeworms, the flukes, and roundworms. They are usually large enough to be seen by the naked eye, though their reproductive form would have to be seen under a microscope. General worm morphology, they're multicellular animals. They have organ systems. They have a reproductive tract that is the most developed. They have a primitive digestive, excretory, nervous, and muscular system. They have thick cuticles for protection. They have mouth glands for breaking down host tissue. Their life cycle would be a fertilized egg, which becomes a larval stage and then an adult stage. The majority of helminths derive nutrients and reproduce sexually inside the host body. Nematodes, so this is one type of parasitic worm. The sexes have different morphologies, in other words, different shapes. The trematodes, the sexes are separate or they are hermaphrodites. which it means that the hermaphrodite means the male and female sex organs are in the same worm. The cestopods type of parasitic worm is generally hermaphrodite. The general life cycle goes from an egg or a larvae to the body of another host, either a different or the same species. There's sometimes an intermediate host involved in the larval development, and then there's a final host in which the adult form occurs. Sources for human infection of parasitic worms can be food, soil, water, infected animals. Routes of infection are oral or the penetration of unbroken skin. Humans are the definitive hosts of many species and the sole biological reservoir for about half of the parasitic diseases of worms. Animals or insect vectors can serve as reservoirs. So some example of helminth disease, intestinal nematodes. They require human host, so does these various ones. They can be ingested, fecal pollution of soil, water with the eggs of the worm, or ingestion of the eggs. The larval form, the Trichinema worm, so uncooked pork can contain this larval stage, and consumption of the not properly cooked pork can contain the larvae. Tissue nematodes, the larvae burrow into the tissue. They can be transmitted by flies. There's a black fly that causes river blindness. There's a guinea worm that can infect humans, usually water that contains the cycloclops. There's
Where do
flatworm.
where do like the worms, like tapeworms and all of that, like where do they usually reside in the human body?
In your digestive tract. Though some of them can get into the lymphatic system and into the blood system. So it depends.
Okay, what
on
happens
the
when they
type.
get into the blood system?
I'm not sure that they can definitely reproduce in your lymph system. I was pretty sure I read, but I'm not sure. Maybe it'll have it about the blood system. So, well, here's one that says schizosoma is a blood fluke and requires either a human or a snail. Can come from ingestion of fresh water containing the larval stage. Here's another a pork tapeworm that can live in humans and swine. Consumption of undercooked or raw pork. A fish tapeworm that lives in the fish, but then can get into humans. That can come from undercooked or raw fish. There's a pinworm. It has a life cycle, a very common infestation of the large intestine. The worm can be 2 to 12 millimeters long. If you remember the thickness of your thumbnail, of your fingernail, sorry, is 1 millimeter. So 2 fingernails thickness would be 12 fingernails stacked would be 12 millimeters. These pinworms are tapered and curved cylindrical shape. They're simple, uncomplicated infection that does not spread beyond the intestine. So pinworms and roundworms, the egg can be swallowed. Eggs emerge from the anus. Scratching can contaminate the hands, and it can be then transferred to a new host. Helminth classification, identification by their shape, size, development of organs, presence of hooks, suckers, other special structures, how they replicate, the type of host they inhabit, whether they, the appearance is either eggs or larvae. There's microscopic identification of the adult worm or the larvae or the eggs. Often have distinct shapes or external, internal structures. Occasionally cultured to verify all the life stages. There are about 50 species of helminth worms distributed in all areas of the world. Some may be geographically restricted, higher instance in tropical areas. Yearly estimate of worldwide cases is in the billions. Conservative estimates of 50 million infections in North America. Humans involved in the constant presence. Only recently have humans evolved, human civilizations got to the point where we can have a helminth-free existence. Absence of helminth infections can actually contribute to autoimmunity and allergy. So, yeah, so this is just a review. Protozoan are always unicellular, no cell wall. The cytoplasm can be divided. They have an endoplasm and an ectoplasm. They can be heterotroph, in other words, different sources of food, or autotroph, a single type of food. Their motility can be flagella, cilia, or pseudopod, or none. And they have the assist life cycle. Fungi can be single cell or multicellular. Cell walls chitin or cellulose. It's chitin. Cytoplasm is not divided. That's only seen in protozoan, some protozoan. The nutritional types for fungi is heterotrophs. They can live on a variety of food sources. Motility is flagella. They can, important structures, they can have those hyphae or spores. Algae can be unicellular, multicellular. The cell wall is cellulose. Cytoplasm is not divided. They can be also heterotrophic or autotrophic. They can have flagella, and they have chloroplast for photosynthesis. Helminths, multicellular, though their reproductive stage can be single cell. They have no cell wall. Cytoplasm is not divided. They are heterotroph. They can have flagella. The ova is the egg here is an important identifying characteristic. Compare that to humans. We are always multicellular. We do not have a cell wall. We do not have divided cytoplasm. We are heterotrophs. Our mobility or motility is limbs. We have no unique structures for identification. So I don't even remember this, the nematodes, cestatodes, and trematodes. In helminths, the most important developed organ system. Sorry, I'd have to go back through the slides. But you can go back if you want to review this. So I'm going to post some material in our first module of summaries of the important slides for the test. And I'll have that posted by tomorrow. I will keep a Zoom meeting open for next week. The test will be about around 75 minutes for exam one. I'll let you know if I have a lecture next week, but you won't be required to go if you're taking the test. I'll just maybe start one of the lectures for an hour and record it. So does anybody have any questions before we go?
No.
No questions? Okay, I'm sorry. That
Yes,
was pretty.
Professor.
Yes,
The exams, how many questions are we expecting?
there'll be around 50 questions on the exam.
Is it all multiple choice?
It will be true, false, and multiple choice.
Okay.
Okay.
Okay, 50 questions. Most of them are true, false, multiple choice. And I'll put in the module 1 a summary of the important slides. Remember, the exam is chapters 1, 3, 4, and 5, not chapter 2. Even though in the McGraw-Hill assignments, I do have a smart book and a quiz for chapter 2, which is on chemistry, which you can review if you would like.
Is the exam going to be on McGraw or is it on Canvas?
You will get to the exam by going to McGraw-Hill campus and clicking on Connect, and then you will see the exam. And when I will send an announcement to remind you of that. That's how you will access the exam.
And it will start at 7.
What I will do is the exam will be open for the entire period of the class, which is 7 to 9:30, but you got to keep in mind that you don't want to start too late. Like if you started at 8:30, you would only have half hour. The exam stops at 9:30. So you want to start early. Start at 7. You could probably even start, if it's 75 minutes, you could start as late as 8. So your window really is starting either at 7, sometime between 7 and 8. Oh, who has the cute baby? All right. So any other questions? Okay, you can always email me or use the Google phone number to text me. And I will send you announcements and watch module 1. I already posted the videos for the first 2 lectures in module 1. Okay.
Okay, Professor.
All right. Thank you, Professor. Have a good night.
All right. Thank you. So good night, everybody.
Good
Stay safe.
night.
Night.